Good afternoon, centers. Yes. How are you? How is everybody? Yes. Shillong, Itanagar, Tamilong, Senapati, Chuachandpur. How are you, children? Good? Done with lunch? Done with lunch? Okay. Fine. Very good. So can we start our class? Good afternoon, Tamilong. Can we start our class? What were we discussing in the last class? I guess we were done with sugars, carbohydrates. Today I was supposedly going to tell you about polysaccharides. That was the only section that was left, isn't it? Polysaccharide part was only left, otherwise we did all the things, isn't it? Tell me. Yes, homework. So I gave you some questions. Okay, one by one, who is going to, please answer me those questions, one by one. So, tell me the question first and accordingly write the answer, because I don't remember the questions in a sequence. So I guess first must be who proposed the double-stranded structure of DNA. That must be the first question. So who proposed the double-stranded structure of DNA? These questions are important, isn't it? These questions are important because these kind of questions will come in your NEET exam. So don't take them too lightly. Yes, double-stranded structure was proposed by Watson and Crick. Very good. And who proved the double-stranded structure of DNA? Who proved it? That proposal is something who is like they are thinking about it without any proof. Same people, is it? Did you study about Rosalind Franklin? A lady who did X-ray crystallography to start the studies about, to confirm the double-stranded structure of DNA. That was proposed by Watson and Crick. So it was Rosalind Franklin. Yes, it was Rosalind Franklin. Exactly, she was a lady. So she did X-ray diffraction studies, and based on those studies, she told that yes, this is a double-stranded structure of DNA. Helical structure of DNA was proved by her. Okay, fine. Semi-conservative nature of DNA replication. Semi-conservative nature of DNA replication. Who did that? Yes, Messerson and Stahel. And what was the experiment they ran? The organism on which they did their experiment? What was the organism they took for their experiment? Messerson and Stahel? Yes, it was proved by Hershey and Chase. Very good. Okay. And how did they prove it? Yes. How did they prove it? How did they prove the double-stranded, the semi-conservative nature of DNA replication? What was the organism they used? Take long breaks, isn't it? Lunch breaks are, good afternoon, Senapati. Lunch breaks are very long. It's 33. Your class is supposedly going to start by 1:15. Okay, purposefully I come a bit late, 5 minutes. And you are more late than that. Please be on time, everybody. Okay, E. coli. Yes, E. coli and bacteriophage, isn't it? So these questions are easy, but yes, when they come at an instance in exam with many things, with many examples, with many names, they do tend to confuse us. Exactly. Transforming principle must be the questions for which you have written F. Griffith. Exactly. So, very good. Nice. Keep it up. Those people who did it, did this homework, very good for them. Those who did not, you should have tried it, because these kind of questions, they do come in exam. So, I'm continuing with the polysaccharide section. We are done with the monosaccharide part. I have taught you the disaccharide part as well. Now, I will be starting with the polysaccharide section. Okay, fine. Let's start with biomolecules and The section under it is polysaccharide. So what are polysaccharide? Poly means many, saccharide means sugar units. So what happens over here is that there are many units which are formed by linking of different glycosidic bonds, and those glycosidic bonds, they give a structure which is called as a polysaccharide. So what is basically polysaccharides? They are comprised of large number of monosaccharide units. So when you take into account many monosaccharide units, they are together bonded by what? They are together bonded by since they have glycosidic bond, they have the name called as glycans. So which bonds they have? They have glycosidic bonds in between them. Clear? Sorted? Keep on copying, children. These things we have done. Okay? Now, next thing I told you yesterday also, one more point, they are either slightly soluble in water, TLY, or they can be insoluble in water. So either they can be slightly soluble in water and or insoluble. Now, the functions, let's see their functions. What functions they do have? They are basically doing what? The functions they are having, what is the particular function? Who can tell me? Yes, function. Function of polysaccharides. They are having nutritive function and they are having structural function. So they serve as a source of nutrients as well as the structures. Fine. Now we'll divide polysaccharides. Did you copy, children? All of you, Tamilnong, Senapati, Shillong, Intanagar. Did you copy this? Okay, very good. Moving ahead. So we'll start up with the first part. Homopolysaccharide. So what is homopolysaccharide? What is it? They are composed of same monomers. Okay. So we'll have some homopolysaccharide. The first one we have is cellulose. Cellulose is composed of many units of beta glucose. How many units? Many means 6,000 to 10,000 units. Huge. They have beta 1,4 linkage. Okay, remember beta? Yesterday we did alpha, we did beta. So if OH is on this side, remember I told you this is beta form, isn't it? So what happens over here? Let's see what kind of structure they are forming. So this is OH again on this side. So they are polymer of what? They are polymer of beta D glucose. Let's see quickly how does this polymerization happens. So there must be a OH over here. So what happens is H2O removal, same condensation process that you have been studying since long. Yes, this is our 4th class of biomolecules, 2nd week running. I need to complete this today. Removal of water molecules, similar way. And what you get to have is, you get to have a bond. It's said to be There's a small mistake. I need to remove this water, not this. Sorry. Sorted. So what is this going to happen? Okay, this is beta D glucose. It's present in cellulose. Okay, this is said to be what? This first carbon, this is fourth carbon. So this is called as beta 1,4 linkage. This one, Shillong? Okay. Clear? Are we clear up to this? Everybody? Yes, children? Okay, very good. Nice. Yes. Itanagar, Senapati, Tamennong. Okay, very good. Senapati, Itanagar, clear? Okay, very good. So this is beta 1,4 linkage. So beta 1,4 linkage is found in which molecule? Beta 1,4 linkage is found in your cellulose. Now next homopolymer let's have. The next, okay, where is cellulose found? Before that we proceed. Where do you find cellulose? Tell me. Where do you find cellulose first and foremost? Where? Plant cell wall? Exactly. So you have two things. You can talk about paper. First thing, talk about wood. And you will see them in cotton too. In wood it is 50% and in cotton it is 90%. Okay? And cellulose is thought to be, or it is the most abundant biomolecule of earth. We know why. Because obviously plants are more in number compared to animals, and all the cell wall of the plants, wood, cotton, everything is having cellulose. So this is the most abundant, this happens to be the most abundant biomolecule on the earth by default. So we have most abundant biomolecule. What is that? That is called to be, is said to be cellulose. Okay. Now the cellulose is also having, or you can say used to form rayon, or what you call is artificial silk. Okay, if you just Google the sites, the shopping sites obviously, and you type in silk saris and all those things, so there will be a particular term called as art silk. This is like marketed as art silk. So they will be like writing in brackets art silk, A-R-T silk. That is, art silk is artificial silk, and that is what? That is basically rayon. And how is rayon formed? Rayon is formed from cellulose. Okay? And what do you use? You use, obviously, this is also used to form paper. So you have all those things you are writing on, not me, you are writing on. I'm writing on digital mode. So this is no wastage of paper, no wastage or no utilization of paper. You are using your paper copies. Okay. So these are the uses of cellulose. Next biomolecule on the list happens to be starch, homopolymer, homopolysaccharide. So what is starch? So this is the stored food in plant. If cellulose was the cell wall, this is the stored food in plants. So what is it doing? Starch happens to be a polymer of alpha glucose okay. Further we talk about starch. They have two types of chains or two types of molecules. It's amylose or amylopectin. So we have two things on list. The first is amylose and second is amylopectin. Clear, children? So who is going to tell me the difference between amylose and amylopectin? It has two types of structures. Amylose is having what kind of bond? They are having alpha 1,4 bond. And this is having alpha 1,6 linkage. Okay? Alpha 1,6, alpha 1,4, amylose. Let's draw quickly to see the alpha 1,4 bond first. 1,4 bond is the same bond that we did yesterday. So this was the same bond. This bond, remember? This was the thing we did yesterday. So this was 1, 2, 3 and 4. And this was 1. So this was alpha 1,4 bond. This is what is called as alpha 1,4. Now, if I'm just extending one more chain and I'm using this particular thing, this was what? This was actually my 6th carbon. Okay. And we have one more unit over here. This is again alpha glucose over here. So this was my 6th carbon. Now again what I do is that I remove a water molecule from here also. Okay, I'm removing a water molecule from here also. Let's see. So this is what more of water molecule from here also. So what I'm going to get is, just I will draw this section, the branch section I'm going to draw. What I'm going to get is, because that section is clear to you already, this is like less of space. Can I move to next page? We have copied this page, everybody. I'm having not much space over here. Just need to explain this on next page. Yes, children, can I copy this? Can I move ahead on the next page? Yes, okay, very good, Shillong. Tamilong, Sinapati, I'm moving ahead. Shall I? Okay, fine. So we have what is now, I'll just draw one bond, one linkage. And what we have is, we will be having CH2OH. OH is removed, sorry. We are having CH2 and the CH2 is basically the carbon thing, because there's one valency left in carbon, so that one valency will be satisfied by oxygen. Okay, and what we have is, we have a very good bond called as, what is that? This is the alpha 1,6 bond. I'm just marking the bond. Okay. This happens to be in between the first carbon and 6th carbon. So this is what is called as alpha 1,6 linkage. Clear? Okay, everybody? Itanagar, Shillong, Sinapati, Tamilong, clear, children? So this is what is amylopectin. Okay, fine. So your NCERT tells that there is a blue color in starch when treated with iodine. I will just add one thing. Since amylose is high in number, it is a single straight chain. It is more in number, so we have prominently blue color more visible. But yes, we need to mention that this gives red color with iodine. We need to mention this thing. That what do you have? This gives, amylose gives blue color with iodine, and this gives red color with iodine. Obviously because of alpha-1,6 linkage. So amylose has somewhat, we could say, around about 200 to 300 units. And this has only 30 units. So huge difference, and that is why you are seeing the blue color. In school you must have done this test and you have seen blue color. So this together gives your starch to have a helical structure. And this helical structure is responsible for holding iodine. Blue, not glue. Blue color. Clear? Everybody, Itanagar, Shillong, Tamilong, Senapati. It's too much of chemistry, I understand. But I hope so it must be clear to you all. So your potato has somewhat, if you talk about it, a different ratio. 80% amylose and 20% amylopectin. Next molecule under homopolymer happens to be the animal starch called as glycogen. What we have is animal starch, it's called as glycogen. Okay, so you have it in liver. Obviously liver has glycogen, muscles have glycogen, but comparative amount in liver is much higher compared to muscles. So what is it? It is a polymer of alpha D glucose. It is D form, better I remove it. Okay, this is a polymer of alpha glucose, and this alpha glucose is basically having both (1,4) and alpha-1,6 linkages. So we have both the linkages in your glycogen molecule, alpha-1,6 as well as alpha-1,4. So this glycogen gives a red color with iodine. So only amylose is the molecule which is giving blue color with iodine. Rest all of them is found in fungi as stored food. So this is something new for us, or you might have been knowing this thing, but they are found in fungi as stored food material. So not only in animals, but yes, in fungi also, the storage of the biomolecule, of the starch component, so you have the carbohydrate component happens to be under this section. Done? So you have studied three molecules, three macromolecules, polysaccharides, said to be homopolysaccharides. Single unit, multiple times repeated. So they are what? They are, first we talked about cellulose, then we talked about starch, and then finally glycogen. And you have one more, that is chitin. So where is chitin found? Quickly tell me. Animal exoskeleton plus fungal cell walls, isn't it? Vinny must have taught you this thing, fungal cell wall. Biology classification is over, I guess. So animal exoskeleton, I have taught you that also. So you know animal, what did I write? Animal. Arthropoda exoskeleton. So if you wonder why crab is so strong, okay, so obviously that is basically chitin. If you wonder why cockroach is having that typical brown shell out of it, brown cover out of on its body, that's typically because of chitin. So chitin is a particular biomolecule which is homopolysaccharide, and this homopolysaccharide gives it a structure where we have some variations. It's a linear polymer of N-acetyl glucosamine bonded by beta 1,4 linkage. So you have what? You have is N-acetyl glucosamine units, many units of which molecule? N-acetyl glucosamine units. We also call it to be NAG, NAG. Remember NAG and NAM as the bacterial cell wall components. Sir must have told you this. NAG and NAM are the bacterial cell wall units which go cross-linking to form the cell wall in bacteria. Okay, so this is what? This is an amino acid derivative. So this is the amino acid derivative of beta glucose. So this is second most abundant biomolecule on earth, following cellulose. Done? Any doubts up to this, everybody? Do we have any doubts up to chitin? Polymer of fructose, I told you this in last class also. And where fructose is having which kind of linkage? Beta 1,2 bond. Okay, this inulin is found in obviously fruits where fructose, but this is abundantly found in dahlia. It's winters, dahlia is coming now. And a plant called as, artichoke. Smallest storage polysaccharide. Clear? Everybody? So we talked about 5 homopolysaccharides. What were they? Cellulose, starch. Then we talked about chitin, glycogen, and finally inulin. So you have 5 different types of biomolecules which are under the category of homopolysaccharides. Now let's discuss about heteropolysaccharides. Not much to discuss. Just write down the names of them so that if they come in exam, you don't get confused. So heteropolysaccharides, we have many of them. We have hyaluronic acid, the first among them. Then we have is chondroitin. Chondroitin is present in cartilage and hyaluronic acid is present many where. Chondroitin. It is present in sperm head also. Hyaluronic acid is present in between the joints also. Then we have is heparin. Okay. You can add on pectin, that is the cell wall of the plant cells. The particular thing, grittiness of the guava is basically because of pectin. If you like chew guava, you have that gritty thing. That grittiness is because of pectin. Then we have, this pectin is also termed as plant cement. Okay, and after that we have is a particular molecule called as hemicellulose, again found in the plant cell wall. Sir must be teaching you plant cell biology. He will be telling you all those things and he will be teaching you cell wall. So hemicellulose, pectin, heparin, chondroitin, hyaluronic acid, these are the particular molecules which come under the category of what? Which come under the category of heteropolysaccharides. So homo is done, hetero is done. Clear? Everybody? Now you have one more thing apart from this. You have is third type of molecule. So this was heteropolysaccharides. This was B section. And we have is nothing much to worry. Mucopolysaccharide. So what is mucopolysaccharide doing? This is particular slimy thing. If you like, bhindi, okra, lady's finger, you know that? Everybody, it must be found at your place. You must be aware about bhindi, I guess. Okay. Everybody, bhindi, lady's finger, okra. Tell me, you know or not? Yes, you know, isn't it? Must be. Yes, it's the national food, I guess. So you must have noticed it's a bit of slimy. Have you noticed? When you cut okra, bhindi or lady's finger, it's a bit of slimy. Okay, that sliminess is because of this mucopolysaccharide kind of thing. So that, just remember, need to remember that particular slimy thing that comes out of the lady's finger when you are cutting it. Obviously, that particular slimy thing is nothing that is but called as mucopolysaccharide. So mucopolysaccharide is what? The slimy polysaccharide. This binds proteins. Obviously proteins and water. Okay. So this in plants they form mucilage. That particular thing you have in bhindi or lady's finger is called as mucilage. So you like bhindi? Yes, everybody? Do you like bhindi? Shillong? No, nobody likes bhindi. Yes? Okay, there are many people who like bhindi. Okay, fine. It's good for health. You should have green vegetables. Okay, nice. Very good. Clear? Have you heard about agar-agar? Your biological classification is over. You know it is obtained from Gracilia, isn't it? Sir must have taught you. It's obtained from Gracilaria. And this Gracilaria, apart from that is mentioned in your book, I'll be giving you 2-3 more terms. It's obtained from Gelidium. It's obtained from Chondrus. This is something new. So it's obtained from 3 varieties of algae. What are they? They're Gracilia, Chondrus and Gelidium. What are they? They're all red algae. So this is all, yes ma'am, okay, fine. This is all about polysaccharides, long I know, but yet it is, they are important. If you just cover these points, you would be able to do almost all the questions from your MCQ books, I guess. Otherwise, let me know if you have any issues, anywhere, any problems, just let me know. Okay. So, we're left with one topic, that is primary and secondary metabolites. After that, I will be starting with enzymes. So, who is going to tell me what are primary and secondary metabolites quickly? Who is going to tell me? Copied this section, this slide? Did you copy this slide, everybody? Shillong, Tamilong, Senapati, Intanagar. Did you copy this, children? Okay. So what we are going to do, we are going to have primary and secondary metabolites. So, primary metabolites. Secondary metabolites. So, primary metabolites are those that are used by your body in production of energy. So, if you are utilizing a particular metabolite for the usage of energy molecule, as energy molecule, for example, if you are using carbohydrates, lipids, proteins, they are sources of what? They are source of energy. So, if you are using any of the molecules as a source of energy, you would call them to a primary metabolite. Obviously, all the primary metabolites are formed by plants. But secondary metabolites are also formed by plants and some fungi. The difference being that they are never used as a source of energy. So let's have a look. They are used as what? Source of energy. They are not source of energy, but what they do is that they help in many other processes. But not source of energy. So, what kind of processes they are helping in? They are helping in many processes. Like for example, we'll talk about some of them. They are some pigments like alkaloids, terpenoids, toxins, drugs. So some of them are pigments. Some of them are pigments. Let's write them all. Alkaloids. We have flavonoids. We have rubber. We have essential oils, isn't it? We have antibiotics, that is fungus. That's why I mentioned fungus over here. When I was telling you, obviously, I did not write it. It was antibiotics. Then we have some scents, gums, some spices. Okay. Did we mention drugs? Drugs. So these are the typical uses of secondary metabolites. And what about primary? They are obviously the energy sources like carbohydrates, proteins, lipids, and all. So they are energy sources, and these are not energy sources, but they obviously act for many things in our body. They are actually used for human welfare. What we use is, because the list what we mentioned is not for their welfare, but we actually try to extract those products and do our own welfare. So they are used for human welfare. Yeah, although these carbohydrates, protein, lipids, they are also used for human welfare, but the plant is also using those resources. You know those things, isn't it? So plant is also using similar resources, and we are also sharing with them. But the secondary metabolites are basically formed by the plants for different mechanisms, for defense, for attracting their pollinators, these kind of activities. But yes, we are using them for what purpose? We are using them solely for our welfare. So these are primary and secondary metabolites. Clear? Yes, children. Everybody Shillong, Tamilong, Senapati, Itanagar. Clear? So all the biomolecules have been done by now. We are left with only enzymes. Can I move ahead with enzymes? I guess I can. Enzymes. So what is the structure the enzymes are representing? They are representing the tertiary structure of protein. What are enzymes? Enzymes are biocatalysts. I guess you know what is biocatalyst. What is a catalyst, you know? You know what is a catalyst? It increases the rate of reaction. So they increase the rate of reaction, but yes, the condition is that they don't get consumed or they don't get totally exhausted in the reaction themselves. So if catalysts are present in chemical reactions, the enzymes, they are biocatalysts present in the biological reactions taking place inside your body. So till now we know that tertiary structure of protein, when we talked about tertiary structure of proteins, we know they are stabilized by many different kinds of bonds, isn't it? Plus, what they are made up of? They are made up of amino acids. So they are stabilized by various types of bonds and they are made up of amino acids. So they might be having a structure similar to this. isn't it? This particular structure is called, this particular part is called as active site. This is an enzyme. And to this active site, we will be having a specific substrate that binds to. This reaction will run in a reversible order since enzyme substrate binding is transient. Transient means it is not like final step. So ES complex is formed. Now this ES complex turns into a different thing. See, the reversible arrow has changed. It is single-sided arrow now. It is called as enzyme product complex. So substrate converts into the respective product. And therefore we can see also here also that there is a specific substrate which is binding over to it. If I am having a substrate of this shape, this is not going to bind to this particular active site. I should have this particular substrate of the shape of pizza slice, and that is only going to bind to that particular enzyme's active site. So this whole pie or whole pizza is not going to bind, but only the pizza slice is going to bind. And this gives you what? This gives you enzyme product complex. So after this enzyme product complex, what happens? Again, reversible arrow will come. Once the enzyme product complex is formed, what happens is you will be releasing the enzyme as such without any changes, without any structural. And then we have is what? We have enzyme plus product complex. So this enzyme, what happens is again ready for next round of binding with its respective specific substrate. So in this way, an enzyme always binds to a substrate, and after binding it gets freed. Clear? So it's very much clear now that what is the catalyst doing? Oh my God, 5 people are sitting on same bench. 5 or 4? 5 people are sitting on same bench. Why, Shillong? 4 people. What is, how many? 4 people are sitting on same bench. Comfortable? Okay, fine. Don't fall off. The girl sitting on the edge, please don't fall off. Make sure you're comfortable. Okay, it's okay, fine. No issues. We also used to do these kind of, okay, it's cold. Okay, nice. So what about those people who are sitting single, like on a bench, single bench? What about those people? Okay, fine. No issues. We also used to do these kind of activities. It's okay. No issues. So this is tertiary structure of protein. You need to remember this. And tertiary structure is stabilized by various types of bonds. These things I have taught you. I have taught you all these things. This is kind of revision. This was easy. This enzyme substrate complex, enzyme product complex, these particular structures you have been studying in surface chemistry also, I know. So if you talk about a catalyst, what is a catalyst basically doing? A catalyst or a biocatalyst is what is it basically doing? So if you talk about an enzyme, what does enzyme do? This increases, or we could say the rate of reaction is changed by an enzyme. How? How would you define a rate of reaction? Rate of reaction is delta P by delta T. Isn't it? This is what is delta P by delta T. That is the amount of product formed in the given unit time is said to be the rate of reaction. So this is how you determine that if your enzyme is having a good rate or not. So if an enzyme is having a good turnover number means formation of product is high. So that turnover number determines, or we can say rate of product formation determines the So if an enzyme is having a high turnover number or the rate of product formation is high with the enzyme, obviously that enzyme would be what? That enzyme would be a more efficient enzyme. So the efficiency of the enzyme is basically determined on the rate of formation of product. These things are very easy. I know you people are getting through all these things. If not, let me know please. Surface chemistry. This is simple surface chemistry till now. I'm moving ahead. I've not moved into biology now till now. Now I will start with the biology section. Okay, but activation energy is left. After activation energy is... Yes. Done children? Everybody? Shillong, Itanagar, Tamilnong, Senapati. Are we clear? Okay, very good. I'll be giving you a break, I remember. But let me finish this chapter first and then we will go for a break. And let's see if after possible, after break I will start with reproduction in organisms. Okay, I had a talk with Vineet sir yesterday. He told that cell division would be done by him. Cell and cell division. He would be doing those chapters since he will be covering your genetics also. So he told that he would be doing cell division section. I will continue with class 12. I will start up class 12 after this. Okay, so your zoology class 11th is over. This is the last chapter we're talking about. Yes, children? Done? Okay. So, who is going to tell me what does an enzyme actually do that makes a reaction faster? Like the fastest enzyme known is, let's see how many of you remember. Carbon dioxide, water, there was an enzyme over here, sorry. H2CO3, what was it? Quickly. And then again, H plus and HCO3 minus. Quickly, what was the enzyme over here and here? Fill in the red blank. What was it? Ah, very good. Keep it up. And where is this reaction going on? Good one, Shillong. Very good. Keep it up. Where is this reaction going on? Where is this reaction going on? Plasma or RBC? In plasma or RBC? Quick. Yes. Very good. Where is this reaction going on, children? RBC. But if you get in option plasma, plasma is also equally correct. But this is more in RBC but less in plasma. Just make a correction to it. Both plasma and RBC have carbonic anhydrase, but the concentration of carbonic anhydrase is higher in RBC but lesser in plasma. Okay, this is the fastest enzyme known. So, what does an enzyme actually, or what is the catalyst actually doing that makes the reaction fast? What is it doing? Is it doing something seriously or not? First thing. Yes. Is it doing something seriously? Or it is as such? Just. We have a thinking that they are doing something. Yeah, they are acting as catalyst. Mechanism I'm asking. I know they are acting as catalyst. We have a long class, a long discussion over that also. But how? The mechanism. Mechanism, the basic theory behind it. No one? Anybody? How does the enzyme increases the rate of reaction? Yes. by number 1, it lowers the activation energy. So enzymes, they actually increase the rate of reaction by number 1, lowering the activation energy. Okay? Number 2, provides a site for breaking of old bonds and making of new bonds. Old bonds are the substrate and new bonds are the product. Okay, just have an example before we are moving ahead. Just assume that you are living on 15th floor. Okay, you are living on 15th floor and you have two options to get to that floor. Obviously, you have a staircase and you have a lift. So, what will you be using? Supposedly going on 15th floor, your house is on 15th floor. So, what would you supposedly use? A lift or stair? Mind it, 15th floor. Quickly, what is making you think so long? Yes, children. Yes, exactly. Shillong? You're in fix. Some people might be wishing to use stairs, I guess. Or some lift. Yes. Senapati, what about you children? Tamilnong? What would you like to use? Senapati and Tamilnong. Shillong is still typing. I guess there's somebody sitting over there, but no. Yes, Shillong, what is making you so long, think so long? Obviously, what would you be using is, what? Lift. Because it depends on number of times you are going ups and downs. Shillong is going to use steps. Now just imagine, you just go back home. Okay, I don't know how many times it happens with you, but you have just entered your home and your mother says, okay, this thing is not in grocery. You have to go and get it. Senapati, the question is whether you would like to use a lift or stairs if your house is on 15th floor, 1515. Yes, Shillong, those people who want to use steps, for them, suppose you are just back to home from your classes and your mother says that these things are not in the grocery. You have to go and get it now. I don't know how many times it happened with you, but this is very frequent at my home. It used to be very frequent at my home. Used to get back and then again go out to get something. Okay. Now what would you do? Again go back with steps, 15th floor, ground floor, and then again come back. Is it? Obviously you would be using lift at some point of time. If obviously once a time, once a week, once a day is okay. But if your frequency of usage is higher, you will use lift, obviously. So what happens is the enzyme is actually a lift. Okay. The enzyme is actually a lift. What enzyme does is, enzyme provides a surface. Okay, everybody look over here. Yes. 4-seater people, look over here. Shillong, Tamilnong, Senapati, please. Itanagar, please pay attention. So what does enzyme do? Enzyme provides a surface. Okay, suppose this, my hand of this is a surface. This is a surface. So enzyme is providing a surface which is actually breaking the old bonds in the substrate and forming new bonds in the product. So when old bonds are broken, if you want to make friendship with somebody who is already in friendship with somebody else, obviously what you would do? You would divide them, isn't it? You'll just break their friendship and then you want to make friends with a person, XYZ person, whosoever it is. So this is the particular surface provided by enzyme where the old bonds are broken, obviously from the substrate, and we get what? We get the new bond formed. So as soon as the new bonds are formed, we said that this is what is called as product. Okay? So you want to break the old bonds and you want to make new bonds. This is where this is taking place on the surface of the enzyme. So this surface actually does two things. Number one, as we have seen in the table, that is lowering the activation energy. It lowers the energy requirement. Obviously, if you are using lift in the place of stairs, you would be using a lesser amount of energy. Plus, this all things happens in a very short span of time. Let's see the next advantage. This takes less time. So it's very much similar to a lift. You would be taking to go to 15th floor, 10th floor, whatever floor you wish. That is what is the lowering of activation energy and taking less time. In what process? In the process of breaking, see over here everybody, breaking old bonds and making new bonds. Clear? So this breaking of old bonds, making of new bonds would be a more energy consuming process, would take more time without enzyme. But if you use enzyme, this whole process is very much sorted, very much easier. Why? Because it provides a surface where we can break the old bonds, make new bonds in comparatively less time, utilization of less energy. Clear? Everybody? All the centers? Shillong? Yes, give me a thumbs up if it's clear. Ah, very good. Nice. Okay. Itanagar? Senapati? Tamilong? Clear, children? Yes. Clear? Very good. Noted these points, everybody? Okay, fine, very good. Senapati? Clear, children? Okay, so let's draw quickly a graph. The substrate. Substrate, sorry, this is a product. On this axis, I'm going to show, not here, mention substrate over here. On this axis, we are going to have the potential energy. Do remember these graphs? The x and y axis markings are quite important. They often are asked, obviously from NCERT only. They are often important because yes, they do ask these things, that where are you going to plot these parameters. So this would be the progress of the reaction, and that would be on the other axis we will have potential energy. So this is what is the particular energy level. So if you talk about the, this energy level is nothing but this is the, you can measure What is this? This is the activation energy. So we have two activation energies. Shall I use a... So this blue thing, blue arrow shows the activation energy without catalyst. So this is a particular option you will be using stairs. Okay. And this is the, obviously, the activation energy with catalyst. So you have two parameters. What are they? Activation energy with catalyst and activation energy without catalyst. Clear? So what kind of reaction is this? Endothermic or exothermic? What is this reaction? Exo or endo? What is this? Endo or exothermic reaction? Quick. Basic chemistry. Very good. Keep it up. So if your P, that is product, your P is at a lower value compared to your substrate, which is in this particular graph, it is an exothermic reaction. That is, you will be getting heat after this reaction. Heat will be released after this reaction. Other way around, if substrate is lower compared to product, that would be an endothermic reaction. So this is what is called as exothermic. This is given you in an NCERT book. You have to do these kind of questions. Okay? So this is what? This is an exothermic reaction. Very good. Who answered this question? Who answered this question? Who was it? Just raise your hand. Shillong. Edney. Very good, Edney. Keep it up. Very nice. I wish I could be there. I would have given you a 5-star for that. Very nice. Okay, fine. So let's move ahead. Or whatever chocolate you like, obviously. So next thing what we have is, we have the factors. What are the factors affecting the rate of reaction? Okay, so things are clear up to this, everybody? How is it? Difficult, easy? How did you find the whole biomolecule chapter? Nice? Bad? How was it? No comments. Shillong, Itanagar, Tamilong. Senapati, how was this chapter? Nice? Moderate? Medium, yes. I guess it must be medium. Okay. So, next thing is the factors affecting the enzyme activity. So, how many factors are you aware about? Factors affecting enzyme activity. So, what are the factors? Yes. Tell me what are the factors you are getting from enzyme activity? Factors affecting enzyme activity. Yes, children? Senapati, where are you? Lost? Are we connected? Not responding since long. Are we connected, children? Okay. So how many of you remember the pH at which saliva acts? Yes, Senapati, not answering since long. What happened, children? All good? You're getting things? Senapati? You getting everything? Okay, very good, Shillong. Yes, Senapati, are you getting, children, everything, whatever is being taught? Any confusions? Since long you're not responding. Everything okay? Okay, fine, very good. Keep on responding so that I could know that you are getting. Okay, fine. Okay, Shillong, very good. 6.8. And what is the particular pH at which HCl activates pepsinogen to turn into pepsin? Yes. Digestion chapter was your test last week. You must be able to answer these questions quickly. pH of HCl at which pepsinogen is converted into pepsin. What is taking so long? 1.8? Sure. It's like variable 1.2 to 1.6. Exactly. Tamil long, just make a correction, small correction. It is variable 1.2 to 1.6. Okay. Okay, fine. Very good. Nice. So, you agree that every enzyme has a particular pH on which it is going to act. Like salivary amylase, you only told me it's 6.8. I asked you, you told me. For HCl to activate pepsinogen into pepsin, that means pepsin is having a particular pH to be active, that is 1.2 to 1.6. That means every enzyme is having what? Every enzyme is having a particular factor called as pH, which is going to affect its rate of process. So if you are altering the pH, obviously the rate of reaction is also going to get altered. How? So this is a pH which is called as an optimum pH. This is what is said to be the optimum pH, at which you have the rate of the reaction. So how would we plot x and y axis? Who doesn't tell me? What is x axis over here? pH is on this plot, obviously, and this is the enzyme activity. These kind of questions, they do come in exam. They would be asking you the particular factors on the x and y axis. So you have to note it that x axis is showing the enzyme activity and y is showing the pH. Sorry, the other way around. Sorry, very sorry, the other way around. X is pH and y axis is showing the enzyme activity. Similarly, second factor, what is affecting the rate of reaction happens to be the temperature. Now, same thing. If we talk about enzymes, we told that enzymes were what? Enzymes are proteins. And proteins, they are having an activity in a particular range of somewhat 25 to 40 degrees Celsius. So if you decrease the temperature, it is going to decrease the rate of reaction 2 times. So if you are decreasing the temperature, how much? 10 degrees Celsius. The rate of reaction decreases by twice, 2-fold decrease. If you increase the temperature 10 degrees Celsius, the rate of reaction increases twice. So an increase and decrease in temperature are going to affect the rate of reaction. Why? Because the enzymes are nothing but they are proteins, with 2 exceptions obviously, that are all enzymes are protein except ribonuclease P and ribozyme. Just make a note of it. So we have ribonuclease P and ribozyme as the two enzymes which are non-proteinaceous. Apart from that, all the enzymes are proteins by their nature. Clear? Yes. Yes, children. Done? Everybody copied up to this? Things are clear? pH temperature role is clear to everybody? Okay, very good, Itanagar. Tamilong, Senapati, Shillong. Nice, very good. So we'll draw a similar kind of graph. So first thing you tell me, why do you store your food in fridge? Why do you store your food in fridge? What happens? When you decrease temperature, like fridge, what is temperature of your fridge? It is somewhat 4 degrees Celsius. Your the particular section in which you have ice, the particular section of forming where ice is formed, that is -4. Okay, the whole fridge has a temperature of 4 degrees Celsius otherwise. Okay, the general temperature is 4 degrees Celsius. So if you are keeping your food, storing your food in fridge, you are actually doing what? You are inactivating the enzymes of microbes. So the enzymes of the microorganisms won't activate at such low temperature, and thus your food is prevented from spoilage. When you increase temperature, obviously I'm talking about more than 40 degrees Celsius, what happens is the enzyme, which are basically what? Which are basically proteins, denature and get their structure distorted. So they used to get their structure distorted, and because of this, what happens is the enzyme loses its activity. So they cannot be reversed. So it won't be able to catalyze any reaction now because it has what? It has got denatured. So once it getting denatured, its whole activity is distorted, whole structure is distorted, and thus it loses its activity. Because you know what? They have active sites. On those active sites, what happens is the particular substrate binding takes place. So if they distort or they damage their active sites somehow, that substrate binding won't take place and enzyme is of no use. Clear? So we will have a similar kind of graph for enzyme activity also, like we did for pH. Similar kind of thing. This is the enzyme activity graph. This is what? This is the temperature optimum. This side we have the activity of enzyme, and this side we have is temperature. So highest enzyme activity will take place at which temperature? At optimum temperature. So what is optimum temperature? Is the best temperature at which the enzyme will act most efficiently. The efficiency of the enzyme is very, very high at this temperature. Clear? Done, Shillong, Itanagar, Tamilong, Senapati. Are we clear up to this? All of you? Yes. Copied is okay, but are you clear? Copied is okay? Yes. Okay, Shillong has... Okay, it's clear and copied. Nice. Okay. Yes, Senapati? Clear, children? Exactly. Okay, very good. The third factor that comes into account is substrate concentration. So how is substrate concentration increasing or affecting the rate of reaction, decreasing the rate of reaction? Let's see. So just for example, you take into account that you are going, like, there are somewhat 50 students in the class. All of the class has 50 students. Okay. Now there is a test, like every week you have a test. There's a test going to be, and we don't know how many students are there. So we got some printouts of the question paper, but the printouts were less in number. Like I first made a printout of 5 questions. So 5 paper. So there were only 50 people are there. So I would be giving 5 question papers among those 50. Isn't it? Not sufficient. So obviously only 5 of you will be getting that question paper, 45 will not. Now I am gradually increasing the number of question papers. I will ask more printouts, ask for more printouts. Now this time again I did not count how many printouts. I told randomly 10 printouts. Again I am just increasing. Let's imagine you are enzyme and the particular question papers are the substrates. Okay, now again the substrates are coming, 10 molecules or 10 question paper of the substrate, question papers are coming. Again, those are given to 10 students. Again, we have some more people, many more people in fact, who are left to have those question papers. Only 15 are saturated with the question paper. Rest 35 are yet to get the question papers. Now again, I increase the question paper concentration, substrate concentration. I ask for 20 more printouts. Again, 20 substrate comes or 20 question paper comes and they are consumed by 20 more students. Again, we have 15 students. So randomly, I am just asking for printouts. This time, I am asking for 30 printouts. Would I be left with printouts? Yes or no? First, I gave 5, then I gave 10, and 15, and 30. Would I be left with printouts this time, last time? I guess I would be. Isn't it? 5, 10, 15, 15, 20. 15 and 20, how much? 35? And then again I asked for 30 printouts more. So obviously I would be left with some printouts, and that is what I am comparing them to substrate. So for a particular point of time, the printouts were coming, they were getting consumed. It were coming, getting consumed. But the last turn was having more of printouts, not getting consumed. Obviously, you won't be taking 2 question papers. It's difficult to do 1. Why would you take 2? Okay. So, we will not take another question paper. We are saturated with 1 question paper. That is the same thing the enzyme is doing. Once it gets a substrate, until unless it is converting it into product, it is not going to leave the substrate. Similar way, once you're getting a question paper, until unless you are answering the whole thing, you will not be letting it out. Obviously, you can't take another also. So, this is the way how the substrate affects the rate of a reaction. Initially, when substrates were coming, when printouts were coming, they were quickly getting consumed because many of you did not have the question paper. Same thing happens with enzyme. If you are increasing the rate of reaction, rate of substrate, increasing the concentration of substrate, initially the rate of reaction will start to increase. Because why? Because the enzymes are getting substrate. But once all the enzymes are saturated with substrate, they will not take any other substrate because their active sites are occupied. So initial increase in substrate concentration increases the rate of reaction, but after a particular time, when substrate concentration is very high, it is not going to affect the rate of reaction. Why? Because the enzymes are still occupied or saturated with substrate. Clear? So this is what is the substrate concentration doing to enzyme activity. Let's see. What happens is, first of all, initial substrate concentration. This is how I'm going to present S. Increases the rate of reaction. But later, no matter how high is the substrate concentration, rate of reaction remains constant. So, why? Why is this happening? Because initially enzymes were not having substrate. We started to increase S. Enzymes started to bind with S. Once all enzymes are saturated with S, any further increase in substrate concentration will not affect the enzyme activity. Clear? So this is what is substrate doing to the enzyme activity. Please copy this. Clear? All of you? Shillong? Senapati? Tamilnong? Itanagar? Clear with this? Clear, children? The funda is clear. How is enzyme affected by different factors? 3 factors only you have to study. Clear? Tell me either it is yes or no. Okay, Shillong can just give me a thumbs up. But other centers, they have to type. Okay, very good. Very nice, keep it up. Senapati, Tamenlong, Itanagar. Busy in copying. We've still some time left for your, yes. Okay, fine. So, okay, nice. Very good. Very good. Keep it up. All of you. Very good. So, shall I proceed with the structure of enzyme now? Okay, proceeding. I guess you must have copied. This was long, I guess. Okay, now enzyme is basically called as what? Enzyme is called as holoenzyme. Very carefully keep on noting things. This is holoenzyme. Now this holoenzyme is made up of 2 parts. It has 2 parts. It is made up of apoenzyme plus it is made up of cofactor. The apoenzyme happens to be the protein part and cofactor happens to be the non-protein part. This is the last topic I am teaching you. Okay, don't worry, the last thing from this chapter. Okay, so what we have is, we have a protein and a non-protein moiety coming together and forming an enzyme called as holoenzyme. Now, if you separate out the non-protein part and expect the enzyme to act in a similar way, it won't. So basic thing is what? The apoenzyme and cofactor, they act in a coordination to bring out our chemical reaction in a greater speed with a lower energy. So there are many instances where people tried to separate cofactor and they wanted to see the enzyme activity. The enzyme did not give that activity. So enzyme activity is always dependent on the cofactor. It is equally important as the protein moiety. We'll divide cofactors into further many sections. Let's see. The cofactor can be of 3 types. The cofactor can be either a prosthetic group, it can be a coenzyme. Just keep in mind, cofactor is the part of non-protein. The cofactor is non-protein part. And coenzyme is a type of cofactor. This is a particular thing where we often get confused. Don't get confused. Cofactor is a non-protein part of enzyme and coenzyme happens to be a type of cofactor. And we have is metal ions. If you just go ahead with the chemical nature, this is organic in its nature. This is also organic. But this happens to be totally, obviously metal ion cannot be organic. This is totally inorganic in its nature. This is tightly bound. This is coenzyme is loosely bound. Loosely bound to protein obviously. and this is having coordination bonds. Okay, so these are three types of cofactors. Let's see once again examples. In case of peroxidase as well as catalase, we have heme as prosthetic group. Somewhat here. In NAD and NADP, we have vitamin niacin as coenzyme. You are aware about this particular enzyme, carboxypeptidases. We have Zn2 plus as metal ion, isn't it? So these are the various types of cofactors we do have to study. Okay, just few examples. Just go ahead and do these things completely. Okay, done? Any doubt up to this? Did you copy the previous slide? You did not copy or you don't have doubt? Itanagar, do you wish to see the previous slide? Copied everything? Okay, very good. Copied and clear. Patent statement from Shillong I am getting. 11th class children also say this thing, same thing. Okay, very good. No doubt? Okay, no doubt. Okay, fine. So wrap up this chapter now. It's done. Done from my side, obviously. The last chapter from 11th standard. So we have done human physiology, 7 chapters. Then we did animal kingdom. We did a structural organization in animals, and this is biomolecules. So next week, I was thinking, having an idea, we will be able to start reproduction today, reproduction in organisms, but somehow we did not manage to. No worries, we will start that on Monday, next Monday. So next Monday, we will be starting with reproduction in organisms. Let me see if I can wrap it fast, because it's a small chapter. Just go back home and pre-read the chapter once. That is not having many difficult things, and you have just given your board exams, all of you. To whichever board you belong, the NCERT has a common content. Okay, so just go back home and read your 12th NCERT. First, all of you, do you have NCERT 12th? All of us, do we have 12th NCERT? Everybody, that's a must. Please get it. It's not very pricey, it's moderate, in fact very cheap. Just manage to get it. And just pre-read the first chapter, organism, reproduction in organisms. Just pre-read it. Okay, we'll start with that chapter in the next class. So, do we have any doubts from biomolecules? Just wait for 2 minutes. I do also have to. Okay, fine. I need to talk to people. That's more important. Just 2 minutes of your. Yes, Itanagar, Shillong, Tamilong, Senapati. Do we have any doubts on this chapter, biomolecules? Oligosaccharide is having, oligo means few. So, what they have is, they have few molecules, few units of monosaccharide. So if monosaccharide is single, it cannot be broken down into more pieces, it would be called as a monosaccharide. When two monosaccharides come together and make a glycosidic bond, it is called as a disaccharide. And when few means more than 2 but below 10, they come together and form bonds, they are called as oligosaccharides. Okay? Tamil long, clear? Okay, you have a homework again today. There is a particular topic in your NCERT. Very good, Shillong. Senapati, nice. Very good, Itanagar, Tamil long. So you have a particular type. Just wait 1 second. You have a particular topic in your NCERT, classification of enzymes. Just read and learn this from your NCERT. The topic, classification of enzymes. Just read it and learn it from your NCERT book. It's topic number 9.12.5. This topic number. Just go back home and do this thing and pre-read the organism, reproduction in organism thing. Okay, fine. Thank you so much, all of you. Please keep on studying. Keep up the good work. See you. Bye-bye.